Hi everyone. Good morning. So we'll try to keep it short. I'm here basically to explain the study case of the highly pathogenic avian influenza for the mood project. 10 or 15 minutes is a bit short to explain what we did during these years, but basically, me and colleagues Marie Cecile, Simon, Marius, we work in two types of avian influenza risk mapping. So the first one was basically to study the conversion events and so to predict the probability of having at least one conversion event at country level as a function of proxy variables for poultry intensification. So for this one, we really wanted or were interested in knowing a bit more about the influence of poultry intensification on avian influenza. The other project was H5NX and H5N1 risk maps, and we wanted to investigate mainly three things. The environmental factors driving the increase of these H5NX and H5N1 cases since 2020, assess potential changes or extensions in their ecological niche, and predict areas at risk using ecological niche models based on pre-2020 occurrence data. So sadly, I won't be presenting this one because I won't simply have time, but to quickly explain to you, we use H5NX and H5N1 avian influenza cases. We retrieved this from HPAI, and we use different covariates because we wanted to understand the situation for poultry and wild birds separately. And also we fit several boosted regression trees models before and after 2020, and the data of after 2020, before and so we have several suitability maps for domestic birds before and after 20, and for wild birds for both H5NX and H5N1 viruses. I invite you to read our preprint. That is, you can easily scan it there. Indeed, we found very interesting results in this research. We see how there is a tendency to change before and after 2020 about the dynamics of these diseases in both poultry and wild birds and the covariates related to this type of birds. But you can read more about this in the preprint, but also the results, the outputs are also present in the Mood platform, so you can explore the maps as you wish. And if you have questions, I will be in the coffee. So we continue with the other objective, that is conversion event. So for some of you might know that the avian influenza or highly pathogenic avian influenza can arise from two different types of mutation. So conversions and reassortment. So conversions are a type of mutation in which low pathogenic avian influenza can acquire virulence factors and then becoming highly pathogenic. And a nice research done by colleagues, Dingra et al., they actually put in very clearly that reassortments and conversions are in two different ecological niche. So we have been focusing on the conversions this time. And well, what about poultry intensification? So the process of identification involves quite a bit of steps, and that mean that we have specialized breeds, faster all in, all out, production cycles, a whole system. And in here, Mario Gilbert and colleagues explained very well that the intensification process, it comes down to 4 pillars. That is higher density, high trade, allele out, and low genetic diversity. And all of these have very clear impacts on the arisal of pathogenic influenza, highly pathogenic influenza. So to quickly explain what we did in here, so we did a literature search and found 49 conversion events from 1961 to 2021. And we use several types of covariates related to intensification, and we fit 2 types of models, GLM, a mixed model, and an inla model Bayesian. These outputs are also present in the Mood platform. You can visit and explore the outputs. So to explain a bit the methods, well, we did a literature review following certain keywords. We retrieve poultry intensification input data it's, for example, chicken, dogs, and turkey poultry density, output input ratio, poultry stock, GDP, etc. At the end, at first we have 32, 33 variables, but after the variable selection procedure, we feed the models with 5 final variables that were poultry stocks that was used actually as the trials of the model, poultry heads per rural population, output input ratio, poultry density, and poultry density per agricultural land. We fit 2 models, as I indicate, and the response variable was count of conversion events in a binomial experiment. So specifically for the inla model, we fit 5 types of model, a base one with random effects, and one with random effects, and a zero-inflated model, because as you can see in the histogram in here, our data is heavily skewed on the zero. And to actually model a spatial inla model, we have to also create a stochastic partial differential equation, a random spatial field. And for that, we needed to construct different type of meshes. We run the models and we select the mesh that was with the best with the best fit. In this case, that was mesh number 1, as indicated there. So the general results is that we have 49 conversion events from 20 countries. Conversion events were predominantly found in chickens and turkeys, and 20 out of these 49 are linked to wild birds, while the majority of these cases were found in commercial poultry farms. Concerning wild birds, we found 4 conversion events in South Africa, Nigeria, Algeria, and Lithuania. So we wanted to see graphically, let's say, what's the situation between conversions and all these poultry intensification measures. How is this moving through time, and is this like really changing or not? So what you're seeing there is basically blue points are the conversion events, countries with the conversion events, and the conditions that of poultry intensification that those conversion events had at that time. And the black points is basically the rest of the countries. As you can see, that cloud of black points is slowly approaching, but you can see it better in this animation that my colleague Jean Artois did wonderfully. So the yellow points are static; those are the conversion events, and you see that that cloud is slowly, the cloud of black points is slowly approaching to the yellow points, indicating that the conditions of poultry intensifications are slowly mimicking the conditions when these conversion events arise, which is not good. So a bit of the results of the GLM in here. We have, of course, a bit of spatial and temporal heterogeneity. More specifically, for this decade, we see that US, South Africa, Australia, and Europe are among the regions with most risk at having a conversion event. The inlet result, well, we finally use the model with the spatial random field, which well, gave the best prediction. And in here, we can actually see that the GLM and inlet has similar predicted regions, US, South Africa, Europe, and China, Australia arise as, yeah, regions of concern. You also see that China is also there in the predicted risk and the upper limit. And with for both models, the poultry output-input ratio was the covariate that had the most, it was the most important variable to describe or to predict accurately the risk of having at least one conversion event. Nonetheless, we have another variable that was marginally significant, that was the poultry heads per row population, so meaning that poultry, the density of poultry is also quite important in here. And as a final result, we wanted to see more graphically what we have, the predicted conversion risk versus density of chicken heads, and the predicted conversion risk, and then the intensive and extensive farming system. So in your left, in my right, you have density of chicken heads in the X and conversion risk in the Y's. And you see basically that the countries or areas with yellow are countries that indeed have a higher conversion risk and higher density of chicken heads. So that it's a good exercise to see the regions at risk. It's the same or something similar when we explore the ratio of intensive and extensive chicken farming systems. We see that areas that we previously disclosed a risky area is actually seen in here, and plus other areas in Africa, Brazil, China, etc. So a bit of a discussion, why more conversion events in intensified poultry systems? So some thoughts. Well, first, surveillance and detection capacity is not homogeneous across countries, so maybe other events, but they are not identified. The conversions occur in intensive poultry that have conditions with high contact rates facilitating these mutations. We have all-in and all-out systems that birds surviving actually these avian influenza outbreaks are immediately culled, and that prevents the selection of natural resistance. Well, research have shown that chickens are more susceptible to this highly pathogenic avian influenza. And vaccination used for highly pathogenic avian influenza following to control outbreaks in some countries may favor the evolution of escape mutants that may convert into highly pathogenic variants. So the results, the conclusions is that the large majority of the detected conversions events took place in high-income countries with knowingly highly intensified poultry production systems. Europe remained the center of conversions, followed by USA. Chicken output-input ratio showed the best predictor. The global trend towards uniform production system correlate with increased conversion events, and that underscores the urgent need for monitoring and surveillance. And in this sense, the take-home message is that the model identifies poultry intensification and specifically output-input ratio as a key predictor. And there is, we should do some emphasis on the need of intensified poultry systems and their role in disease emergence globally. With this, I would like to take less than 1 minute to, thanks my colleagues from the Spell Lab, my supervisor Simon, Marie-Cécile, Diana, Marius, people from Avia, Guy, Cedric, Tom, Sarah, Wim from ITM, Willie from Ergo, and of course all the wonderful colleagues of Mood, because a project is as good as the colleagues that integrate it. So with this, I finish my presentation. Thank you.
Thank you.